Okay, when you're doing biological experiments, you often need to be really careful of contamination. So to avoid contamination, biologists try to use as much as possible sterile technique. All right, so why are we concerned with contamination? Well, if you're trying to study bacteria on a plate, you don't want other bacteria from the air growing on that plate instead. If you're trying to do a very technical experiment like a PCR, you don't want DNA from your skin getting in your tube and ruining that experiment. So where can contamination come from? Unfortunately, the answer is pretty much anywhere and everywhere. So in the air, there can be bacteria, fungal spores floating and coming down and sort of landing on your work area. All the surfaces, like the benches, whatnot, you can also assume are contaminated. We are a source of contamination, so our hands that we use to do our experiments might carry viruses, bacteria, whatnot on them, as well as our own skin cells. And as I'm talking, all the bacteria from my mouth are kind of coming out, going into the air, and could also come down and land on my experiment. Also, your experiments can contaminate your other experiments. If I have a control plate with one type of bacteria and another plate with a different type of bacteria, I want to be really careful, right? Obviously, I want to, any equipment that I'm using here and then using here, I want to sterilize in between. All right, so how do we minimize chances of contamination? And I'm just going to go over general techniques today. In certain cases, you'll want to use additional sterile techniques. But basic lab practices would be to wash your hands when you come into lab, put gloves on, and even something this simple, there's a technique for it. So when you grab gloves from the box, you want to try to avoid touching the fingers. They're in the box, they're sterile. But once you start touching them, well, any contamination on your hands, you're transferring to the gloves. So try to grab them from the wrist area. Put one on, and now, right, I want to avoid getting these gloves dirty once they're on. And note that gloves come in different sizes. Make sure that you pick a size that fits tightly, right? You don't want gloves hanging off and sort of getting in the way when you're trying to do experiments and using your hands, but not so tight that they're really uncomfortable. Okay, and I'll show you how to take the gloves off in a sterile manner at the end of this. Okay, so before you start working, it's a good idea to take some ethanol, spray your area with these little squirt bottles, wipe it down with a towel to help sterilize whatever contamination might be on here from whoever was working here previously. Also, when you're done working, it would be good to also wipe it down and avoid any contaminants on here from sort of being there for the next person that'll come in. Also, along the lines of keeping your work area clean, you'll have these little trash bins. You want to empty these out at the end of every lab. Okay, when you're working, you want to not talk, right? If I have an open plate here, I don't want to be talking and spreading my bacteria so that it can come and land on that plate. Similarly, you want to minimize air exposure. So when you want to open your plates, close them as quickly as possible. Keep your pipette tips with the tops on whenever you're not actively using them. Same for tubes. Whatever reagents we try to consider sterile, you want to minimize their air exposure. So keep them covered, lids on, etc. When you're not directly using them. And try to work quickly also to minimize that air exposure time. Okay. Later on, you'll probably want to use other things for more sophisticated techniques. You might need to work in the fume hood, or you might need to use an open flame to create an updraft of your work area or help sterilize certain equipment. But for now, just bear in mind gloves, cleaning your surface, emptying the trash, working quickly, keeping things closed as much as possible, not talking while you're working. Additionally, right at the end of lab, you should take your gloves off in a clean manner. So use one gloved hand to grab by the wrist, pull up. So now what's on my fingertips on this hand now is the inside of the glove, right? So that was touching my skin. It was not touching the bacteria or whatnot I was working with. It's clean. I can use that. Grab my other wrist strap, fold them up. Now only the clean area of the gloves are exposed, so I can touch them with my bare hands, put them in biohazard trash, right, assuming I was working with biohazards. And then before I leave lab, I should, of course, wash my hands.